I am very honored to be here today. I am also just a humble biologist, so I will not attempt to span topics as broad as autopoiesis up to human rationality. But I am hoping that I can address this topic from a biological perspective, the biology of human nature, and to point out some of the major problems that exist in this field from a biological perspective. So I'd like to start by making an important distinction between human cells and human beings. I think this is, it seems very obvious to most of us when we look at mature adults who have many trillions of cells. But when we start at the beginning with a human being who is one cell, it's a very important distinction to make clearly. I'd like to then progress to the difference between a cellular soul and a human soul. Uh, Talk a little bit about human individuals with a focus on human twinning, which has been a major stumbling block both for philosophers and for people who have promulgated science policy over the last 50 years. And perhaps if I have a little bit of time at the end to raise the unique issues that are presented by conjoined twins. So human beings versus human cells. We know that an individual human arises from the fusion of specialized cells, the sperm and the egg cell. And therefore, in considering the biologic nature of human individuals, it's important to distinguish between human cells and human beings. And to get us oriented on this question, I'm going to start with a movie from a fertility clinic. What you see in this picture is a series of concentric circles. So this is a human one-celled embryo maintained in a laboratory culture dish. I got this movie from an assisted reproductive website. So the bright circle on the outside is the culture dish. The kind of greeny circle on the inside is the human one-celled embryo or zygote. And it's surrounded by a protein coat called the zona pellucida, or the clear zone, that serves as a protective layer for the embryo during the first 5 to 8 days of development. So I'm going to play a little video, and what you can see from this video is that the one-celled embryo pretty quickly, within the first 24 hours, enters into a phase of cell division without growth. So it's simply taking the matter that it's comprised of and dividing it into smaller cells. Those smaller cells are known as blastomeres. And after about the third round of cell division, we reach a stage called the morula stage, because the embryo sort of looks like a raspberry. And at this stage, an important feature happens. It's called compaction. So the appearance of the embryo changes a little bit. It smooths out somewhat. And this is because the cells, those blastomeres, have developed tight connections between them, each other. And that allows them to transport fluid to the inside of the embryo, where the whole structure blows up kind of like a fluid-filled soccer ball, to reach the blastocyst stage at about 5 days. This is an important stage for development because it's the first point at which we have cells that do different things during the subsequent development of the embryo. The cells on the outside of the soccer ball, the black and white hexagons, are known as the trophoblast, and they're going to largely contribute to the placenta. Whereas a smaller group of cells on the inside, poetically known as the ICM, or inner cell mass, is going to contribute most of the cells into the body that you're carrying around today. So all these events happen as the embryo is traveling down from the ovaries to the woman's uterus. So this cartoon shows, get the right button here. Eggs are matured in the ovaries. They're ovulated out into the fallopian tubes, and all the events we saw in the movie, the formation of the morula, the blastocyst, happen as the embryo is traveling down to the uterus. And at about somewhere between 6 and 10 days, it will implant into the wall of the uterus. Subsequently, the cells of that inner cell mass here are going to divide into 2 layers. And the cells in the upper layer are going to undergo a process known as primitive streak formation or gastrulation. This is going to be important because it's a point at which the most primitive structures of the nervous system arise. So 5 days. 5 to 7 days. So this tells us what happens in the beginning of human development, but it doesn't really answer our question of the difference between a human cell and a human being. So is the product of sperm egg fusion simply a modified gamete? Perhaps many people refer to it as a fertilized egg. Or is it a new kind of human cell that's not a gamete? Or is it a new human person? So we can address the modified gamete versus new kind of cell question pretty simply by considering the scientific definition of when we get a new cell type, either in the laboratory or in a developing entity. So changes in cell type are universally detected by only two very simple criteria, either changes in material composition, so what a cell is made out of. Typically, this reflects a change in gene utilization, or by changes in cell behavior. And very often, those two things go together. If you change the toolbox that the cell has available to it, you can change the kinds of behaviors the cell is capable of demonstrating. Logic would also require that if a new cell arises from the one or more preceding cells, those pre-existing cells have to go out of existence before we can have a new cell type. So, based on that, I think we can confidently conclude that a new cell forms at sperm egg fusion. So in this picture, you're seeing a scanning electron micrograph of a larger pink egg cell and a smaller sperm cell that are in the process of fusing. And upon sperm egg binding, which is the attachment of the outer surface of the sperm to the outer surface of the egg, those two surfaces, the plasma membranes, will fuse in a very rapid and well-studied procedure that takes less than a quarter of a second to complete. So there is an instant, 250 milliseconds, during which we go from two cells to a single cell. So if anyone tells you there's no such thing as the moment of fertilization, that would be it. So in this process of fusion, the sperm cell ruptures and it ejects all of its internal contents, including its nucleus. And also, so it clearly ceases to be. And also, the surface of this fused cell will contain all of the lipid and protein that used to be uniquely associated with the surface of the sperm. So the material composition of the cell has changed due to the incorporation of this material from the sperm cell. So what about the egg? So following the fusion and destruction of the sperm, the resulting cell has a new composition that's distinct from the egg. We've already talked about the incorporation of sperm-derived lipid and protein, but the changes are much more extensive than that. The cell also has a new genetic composition, with half of its DNA being derived from mom and half from dad. And perhaps more dramatically, this cell also radically changes its behavior to act in a very un-egg-like manner. So the entire raison d'etre of being an egg cell is to find a sperm and to fuse to it. And yet within 1 to 3 seconds following this membrane fusion event, this cell will initiate a molecular cascade that over the next 30 minutes will result in the deposition of enzymes out into the space between the zona pellucida, that protein coat, and the surface of this cell. And the function of those enzymes is to destroy all of the remaining sperm binding sites. So the entire raison d'etre of being an egg is to fuse to a sperm, and yet the very first act of this new cell is to antagonize the function of the gametes from which it's derived. So it has clearly entered into a new pattern of behavior that is not consistent with the function of a gamete. And the egg cell, therefore, as a haploid gamete that's designed to fuse to a sperm, also ceases to exist. So a new cell with unique genes and composition, molecular composition and behavior arises at this instant of sperm egg fusion. But what is this new cell? I would define it as a living being, which is an entity composed of parts that mediate self-referential, or that is internally directed, activities that are sufficient to constitute a unity or a whole that is capable of sustaining itself and thereby opposing entropy. So this would be a definition very similar to the definition of autopoiesis that we heard from Dr. DeHaan in the prior talk. But is this simply a human cell, or is it a new human being? I mean, its scientific name is a zygote, and yet is the zygote an individual person? Or is it just a cell, or perhaps a ball of cells? And the difficulty in making that distinction for us is that we don't have a lot of personal experience with embryos at early stages of development. Even I, as an embryologist, can't look at these pictures and go, oh, look, it's a baby. Yeah, I don't have that emotional response. So at early stages, recognizable human form and human functions are not evident. So how do we know that a zygote or a morula or a blastocyst is a human person and not just merely a human cell or a ball of cells? And this is a surprisingly difficult question to answer for a number of very important reasons, the most important of which is this one. All cells are also living beings. I'm going to give you just one example. So this is a film that was taken in 1958 by a faculty member at Vanderbilt University. He took a blood sample, put it in a culture dish, and you're going to see 3 cellular actors in this film. The star of our movie is this cell in the middle. That is a human neutrophil. So its job in the body is to seek out bacteria and destroy them. The other sort of passive supporting characters are these round cells, which are blood cells, and they don't do much. And the other active, at least in this movie, and the other active players are these guys here. So those are some bacterial cells that contaminated the culture. And this movie will demonstrate that cells exhibit autonomously coordinated activity of parts for the good of the thing as a whole. So cells are actually organisms. Let me make the movie go. Oops, here it is. So you can see the cell has internal parts that you can visualize. It's capable of sensing its environment, avoiding obstacles, chasing its prey, doing the things it would naturally do in the body. It's very persistent as well. The suspense is killing me. Oh, it gets it in the end. But the fact that it is doing this with just the bacteria, it's ignoring the other blood cells that are present, shows you that activities that are specific to the function of this kind of cell persist in the absence of any living human being controlling them. The soul of this experimenter was not reaching out into the culture dish to direct the activity of this neutrophil. And it's important to note that activities that can be explained by the properties of cells are not sufficient to conclude to the presence of a human being. And if you take one thing home from this talk, this is the thing I'd like you to take home. The central problem thinking about human nature and human individuality is that both cells and human beings are alive and human, but human beings are not just a collection of cells. So how then do we tell the difference? Well, we got to get a little bit more granular. So this is a very excellent paper written by Bernard Rosenbach. It's about 80 pages, so it would be a very dense, granular analysis of what constitutes a human organism, what goes into that autopoietic processes of self-regulation that allow you to be a living being. So Rosenbach defines 10 distinct properties, and I'm going to try to hit the golden mean between simplistic and complex by breaking these into 4 general classes. So I would say one of them is autonomy, or the ability to regulate internal processes in a manner that allows for the cell to be maintained. And this would include things like perturbations, response to perturbations through wound healing. Integration is the fact that the various parts and subsystems of a living being have to talk to each other in a way that allows for the persistence of a healthy state. Adaptation is similar to autonomy. It's the ability to respond to perturbations that arise from the outside of the entity and adjust to those perturbations in a manner that maintains the health and integrity of the organism. And then development, or growth to a characteristic mature form. So based on that, what is a living being? I think everyone would agree that this is a living being. It has parts, the parts work together for the good of the whole. It exhibits all of the functions I just listed. I think even some of our opponents in a Christian view of human life would acknowledge that this is a human being, or this is a living being, a living organism. It has parts, those parts work together in a coordinated way. But is this a human organism? Is it a living human being? And if so, then why is this not a living human being? So if we go to those 4 categories of characteristics of living beings, the zygote exhibits autonomy and wound healing, and yet so does a neutrophil. Zygote exhibits integration, and so does a neutrophil. Adaptation, but so does a neutrophil. The one thing the zygote does that a neutrophil does not is this one. So it's not just the hubris of me as a developmental biologist saying developmental biology is the central defining element of human nature. It really is. It's the one thing we do as embryos, as a zygote, that's different from what a cell does. And you can see it in these pictures. These are pictures of actual embryos from the Carnegie collection. I am not endorsing the manner in which these images were obtained. But you can see this represents the first 8 weeks of human life, where you go from a single little dot here, that's the zygote, blown up here so you can actually see it. And in 8 short weeks, you progress directly to a fully formed, albeit very small, this is only 5 millimeters here, human form. So the period of embryogenesis is the period in which all the tissues, cells, structures, and organs of your body are formed. So it's really miraculous that this takes only short 8 weeks to do. The rest of prenatal life is the period of fetogenesis, where the embryo or the fetus at this point will grow incredibly in size. The organs and tissues will mature in terms of their function and their biochemistry, but you're not going to make anything new. So entities that are capable of development are by definition multicellular organisms, such as a human being, and they're uniquely capable of development or the self-directed maturation to a characteristic form. Autonomy, integration, and adaptation exist to preserve the healthy function of the multicellular whole rather than the cell as a cell. And from sperm egg fusion onward, the actions of the zygote are only intelligible as actions that anticipate the needs of a maturing multicellular individual. So those of you who are afraid of science can close your eyes for the next few slides. And just remember this conclusion. You can trust Dr. Condic. But I am going to give you a few examples from the first few minutes of human life. So this is a cartoon showing the state of affairs directly after the fusion of those outer plasma membranes in that 250 milliseconds. So we have a nucleus that's derived from the sperm, shown in blue, a nucleus derived from the egg, shown in yellow, and you notice they have different numbers and letters on them. The sperm nucleus is already haploid, so it has half the natural amount of DNA that you would find in a mature human cell. The egg-derived nucleus at this point is still diploid, pseudodiploid, but it contains twice the amount of DNA that it should. Okay, so at fusion, sperm and egg cease to be, and they form a new cell with unique composition and behavior. Many people will assert that the egg cell drives development, but this is factually complete nonsense because we know that factors from the sperm, for example, are required for the egg cell to complete a reduction division that will reduce it to a haploid nucleus. And therefore, each of those nuclei will contribute half of the DNA to the individual. So within 1 to 3 minutes, sperm-derived factors cause a rise in intracellular calcium, in addition to inducing the egg nucleus to enter into meiosis. That's going to give rise to this cortical reaction we talked about that will destroy all the remaining sperm binding sites. Now, completing meiosis, so taking the egg nucleus from 2N to 1N, and blocking additional sperm binding are really not required for the cell to remain a healthy cell. In fact, many, many cells in our body have plenty of copies of DNA, and they're perfectly healthy, the most well-known of which are muscle cells. So this is a histological section of mature human muscle, and all the little black dots that you're seeing within a single cell, 1, 2, 3, 4, 5, 6, 7, 8, those are all nuclei. So this cell has multiple copies of DNA, and it functions perfectly fine. So there's absolutely no reason for a cell as a cell to get rid of extra DNA. So why does it do it? Because it's anticipating the needs of the developing embryo. This is from a recent review. If we don't do these things, if we don't reduce the egg nucleus to 1N, we end up with a triploid embryo. And triploidy is one of the most common chromosomal abnormalities in human gestation, present in 1 to 2% of all pregnancies. However, triploid pregnancies rarely come to term, and the very diverse phenotypes that are seen are all grossly abnormal. So a triploid state is not compatible with development as an embryo, despite the fact that it's a perfectly healthy state for a cell. And so the absolute first act of the zygote is to anticipate the needs of the embryo, the needs, its own needs. So within 30 minutes, meiosis II, which was initiated by sperm-derived factors, is complete. So we now have the correct amount of DNA inside the cell. Zygote-derived enzymes have destroyed all additional sperm binding sites, so we can't add any more DNA to the cell, which would be very unhealthy for the embryo. We're going to change the structure of the sperm-derived nucleus by adding histone H3, which is a DNA binding protein. And the only reason that's necessary is because we can't use the DNA inside the sperm nucleus unless we do this. So again, we're anticipating the developmental needs of the embryo. And we're going to modify the DNA of both nuclei to make them available for transcription, for utilization by the embryo. And this is, again, only required for development. So DNA reduction and differential modification are not required for healthy cellular life, but are essential for normal embryonic development. And there are a large number of these events that occur within the first 10 hours. If you're interested, you can search my name, When Does Life Begin? And it's a freely available paper online that goes through all of these events in great detail. But it's clear from these events that sperm egg fusion sets off a molecular cascade that controls events that take place days, hours, weeks, even months later in life. And that these events are not required for cellular health, but they only make sense as part of a developmental sequence that's produced by an organism. And while some scientists may dislike or deny this conclusion, it's supported by hundreds of peer-reviewed papers, and it's entirely uncontested in the literature. I have made this assertion hundreds of times to scientific audiences, and I've always asked them, hit me with your best shot. Bring me a paper, any paper, published anywhere, at any time, by anyone, that has the tiniest shred of data that contradicts this conclusion. And while you can find many, many assertions by scientists that nobody knows when life begins, there is not a single piece of data out there to suggest that this conclusion is not correct. So, zygotes are unambiguously human beings, not human cells. So, a new cell of the zygote with unique genes and behavior arises in this instant of sperm egg fusion. It immediately initiates a molecular cascade that exclusively serves a developmental purpose. And therefore, the zygote is not merely a modified gamete or a new human cell, but it's a new human individual, or human life begins at this instant of sperm egg fusion. Okay, so now this brings us to the question of a soul. So what causes a human being to be different from a human cell? The Church teaches that a rational soul is a unique form of the body, the Council of Vienne in 1311, Fifth Lateran Council and Brief of Pius IX. The Catechism of the Catholic Church states that the unity of body and soul is so profound that one has to consider the soul to be the form of the body, i.e., it is because of the spiritual soul that the body made of matter becomes a living human body. Spirit and matter in man are not two natures, unified, but are rather their union forms a single nature. So what does this mean for our understanding of the difference between human cells and human beings? I would argue, and as I said, humble biologist, I am not a theologian, and I will be happy to be corrected. But I would argue that the substantial form of the soul causes all acts of a living being. The soul is the organizing principle of the body, and it causes all bodily activities, including life. And all living beings, bacteria, cells, plants, animals, all have souls, but the human soul is immortal. And it's intrinsically united to the body in that all acts of the soul must manifest as bodily acts. So even the act of thinking manifests as a movement or an action within the body. Cells removed from the body, like our little neutrophil, or after the death of a person, undergo a substantial change and obtain a new kind of soul, a cellular soul. I don't have a good name for this soul because it is human. It is a soul, but it isn't a human soul. But what exactly is a soul? These are the kinds of things that keep biologists awake at night. I mean, how do you even understand what this is? So my brother and I wrote a book. This is the book that won the Expanded Reason Award, and I'm going to quote from it fairly extensively, so don't be terrified when a lot of text shows up. So, from a biological perspective, the simplest way to consider the substantial form is to ask, what, in concrete terms, makes a living human different from a simple pile of organic molecules that is identical to those found in the body? What differs between a human and a pile of chemicals is not what they're made of, but how the material is organized or formed. And substantial form is the cause of the observed organization. The precise relationships and interactions among our molecular parts do not arise from the parts themselves, otherwise, there would be no difference between a human and a pile of molecules identical in composition to a human being. Further, the matter that composes a human is ordered by a specific set of rules that's distinct from the rules ordering those exact same molecules in a non living pile. A developmental biologist sees these rules in terms of practical matters of ligand binding affinities or concentration gradients or intracellular diffusion constants and other principles that govern how molecules function within cells and how cells communicate with each other during the formation of a mature body. Yet collectively, the rules or principles governing life, function, and development of a particular living being constitute the substantial form of that individual. Importantly, there's nothing mystical about the substantial form for most living beings. If it were technically possible for an experimenter to start with a collection of inert organic compounds and establish all of the interrelationships between those molecules, cells, and tissues and structures that are observed in a living body, this would create a soul that would subsequently be the cause of all the ongoing properties of the life observed in the entity thus constituted. Now, I will give you a disclaimer regarding theology. While all living entities possess an organizing principle or a soul, the human soul is unique in that it's immortal and therefore created by God. In contrast, the souls of non-human living organisms, while immaterial, are not immortal and therefore can be generated by natural processes or theoretically by an experimenter. So for the remainder of my talk, until we get to twinning, I'm going to talk about non-human living organisms. So they're all living beings, and the soul would be the ordered relationships among the parts that causes those parts to constitute a unity. And life would be the activities of such living beings ordered by the soul. But what might a scientific description of a soul look like? So there's a large field of biology known as systems biology, the goals of which are to define every conceivable interaction between every minute part of a living system and how all those parts interact. So generating, therefore, an interactome. And I think the interactome for many organisms is an imperfect but a first-ordered approximation of the soul of that entity. So this is a paper looking at the interactome of a bacteria. You get these lovely detailed kinds of diagrams, each dot of which represents a particular molecule, and it's connected by lines to all of the other molecules that it interacts with. Of course, human cells are even more complicated. So this is the interactome of a human cell. In this study, they documented interacting partners for 2,594 human proteins in a human cell line. And the resulting network contained 23,744 interactions among 7,668 proteins. So the soul of a multicellular individual this is the soul of a cell, a eukaryotic cell, would comprise all of the molecular interactomes of all of the cell types at all stages of development, as well as the interactions of structures within cells and of all the cell-cell interactions. So you can imagine how enormously complex that would be. What are my conclusions about human souls and cellular souls? So all living beings possess a substantial form or a soul. The souls of human beings are immortal, created by God, but other souls can arise by natural means and could theoretically be created in a laboratory. In biological terms, the soul is proposed to be the ordered relation among parts that cause those parts to constitute unity. And for living cells, the interactome is a first order approximation of the soul. And this raises some interesting questions, which I would love to get some feedback on. So, how then do we interpret cases of life support where aspects of the soul are performed by machines? In such situations or others, can a human soul be partial or diminished? And how then can we distinguish between limitations in matter and a non human soul, given that we know the cells that comprise our body can transition to cellular souls? And at some point in the process of death, the human soul departs, and we have left an aggregate of non human souls, but whose properties of those cells remain robustly consistent, just as that neutrophil did. This is not a trick test. I do not have answers to these questions. All right, human individuals, what about twins? So twins have long been a subject of myth and mystery. Here I am in Rome. This is Romulus and Remus, of course. They've also driven research and policy in very important ways. So starting with a paper by Clifford Grobstein, where he argued that the stages involved, so this was in response to the newly promulgated practice of in vitro fertilization. The stages involved in IVF are not only pre-persons, but are also pre-embryos. They are pre-embryos because separated cells from early cleavage stages of eggs and non-human mammals, and most likely of human eggs, can each give rise to a whole embryo. So, i.e., we can have twinning if we separate the cells of early embryos. And because clutches of cells can be deleted and two clutches can be combined, a single embryo can still result. And therefore, in cellular stages prior to implantation, a single embryo has not yet been established. This argument was taken up by the Warnock Commission in the UK that asserted that at the time of cleavage, at a 2 or 4 cell stage embryo, each cell retains its totipotential capacity, i.e., it could become a twin, a full individual, if separated. Thus, if separation occurs at the 2 cell stage, each may develop to form a separate embryo. And such a separation would lead to identical twins. And therefore, a human embryo cannot be thought of as a person or even as a potential person. It's simply a collection of cells. So the Warnock Commission set the policy for the 14-day rule in embryo research in the UK, which was also adopted in the United States. And it defines the problem of twinning pretty clearly. So if we have a single cell, a zygote that divides to produce 2 identical totipotent cells, each of which, if separated, can develop into a separate individual, how can the original zygote be considered a single human person? So this was the science side of it, but this was also a very big problem for a lot of philosophers. So Elizabeth Anscombe, addressing the possibility that identical twinning involves a splitting of a pre-existing human, similar to the division of an amoeba at mitosis, says, but the obvious objection is that in the case of an amoeba, there's no doubt that we start with one amoeba and it splits into 2 amoebae. But in the human case, it is precisely the question of whether what we start with is a human being. This was an objection also voiced by Norman Ford in his book, When Did I Begin? Due to the possibility of twinning, Ford argues in 1988 that early embryonic human cells could not constitute an actual human individual. Instead, they would have to have the potency to form one or more human individuals. And it seems that the biological evidence leads to the philosophical conclusion that a human individual, our youngest neighbor and member of the human community, begins at the primitive streak stage, at that stage when the earliest rudiment of the neuter system comes about, and twinning is no longer possible, and not prior to it. Joseph Donsiel, also a Jesuit in a very influential paper, argued That the trouble is that if all cells could develop into a separate individual, then every single cell of the zygote, the morula, or the blastula is a human person. For at the earliest stages of embryogeny, each cell resulting from the division of fecundicated ovum possesses such a power and virtuality, and all of the cells are totipotent, each of them, of the many, if separated early enough from the others, could turn into a human being. So, my brother and I tried to address this a bit in our book, but I actually wrote an entire book on twinning that's shown here. And I'm going to just address the objections of these three philosophers. The central issue for them is potency. So if a zygote is a person in virtue of the fact that it has the potential to develop into a mature human, then when all cells that have this potential would be equally human persons. A zygote that has the potential to be two persons cannot be simultaneously considered a single person. So this is the argument of these three philosophers and others. But I think they're all confused in exactly the same way. Potency has several meanings. And here I'm probably preaching to the choir, but I'll make it brief. Aristotle considers potency along two distinct axes, active and passive, and proximate and remote. Let's just consider active and passive. So an active potency is the power to act in a specific manner that's intrinsic to the thing itself. So I have, though it may be hard to believe, the active potency to speech even when I'm currently silent, a situation that has never occurred when I also retain consciousness. Passive potency, in contrast, is the power to be acted upon in a specific manner. So wood has the potency to be fashioned into a chair or a crucifix by the action of a carpenter, and water does not. And neither has the active potency to become a chair on its own. So what about proximate and remote? Proximate potency is the power to act or to be acted upon that can be exercised in an immediate manner. So I have the proximate potency of speech, even if I'm silent. A remote potency is a power to act or to be acted upon in a specific manner that requires some intervening event before it can be exercised. So an infant has the remote potency to speak, but it has to learn how to do it first. And a sapling has the remote potency to be fashioned into a chair, but it must grow to be a tree first with wood and bark. So the kinds of potency that exist in development, a zygote has the proximate active potency to be a human being because the zygote, and therefore the zygote is a human being now because it actively is exercising an intrinsic power of maturation and development. On its own, in an appropriate environment, it will go on to produce all the structures it needs to be a more mature individual of its same kind, of its same species. Early blastomeres have the remote active potency to be a human being, which means that a blastomere now is a part of a single human being, but it can become an independent human being if an intervening event, the isolation of the blastomere, occurs. So the remote potency of a blastomere means that there's a single unified human prior to twinning, and that blastomeres must cease to be a part to actualize their remote potency to be a whole on their own, which means they must undergo a substantial change in order to become a human being. So we never have a zygote that has the potency to be two people. We have cells with remote potency to be a single individual, but they must undergo a change in order to actualize that potency. So the timing of twinning matters. If we start with a mature individual, that Angelicum says is we know already it's an amoeba. We know already this is a mature woman. And we twin her through the process of cloning. So I take some skin cells and I introduce them into an oocyte and we grow it up to be a little mini me. This does not call the identity of the mature individual into question because it's already established itself as a mature individual. But the difficulty thinking about twinning at early stages of development is if each of these cells can go on to produce a single individual, then what's the nature of the first one? So there's substantial evidence that this really never happens. And I've already given you substantial evidence to suggest or to indicate that the zygote is, in fact, functioning as an organism from the very, very beginning. But just to calm people's minds, despite the incidence of monozygotic twinning in IVF being substantially elevated, it's never been observed. No one has ever seen twinning happen at the 2-cell stage in humans. With one report stating that although we've never observed an embryo splitting in half before the blastocyst stage on its own initiative in over 15 years of laboratory experience. And this is likely to be the case because early blastomers are very, very sticky and are strongly inclined to act in a unified way as parts of a single entity. This has been directly tested in, I think, a very elegant experiment where these experimenters induced the formation of twin fraternal embryos, inside a single zona pellucida. So you can see 2 cells inside this zona, and each of them are a fraternal twin of each other, or they're related to each other. And what happens if you let these twins inside a zona go forward is that in all cases, over 200 cases, they observed the twin embryos fused at compaction when the cells developed this highly adhesive state, and they formed a single chimeric embryo. Okay. So twinning at this stage is highly unlikely, and when does monozygotic twinning likely occur? So it likely occurs here at hatching. So we're going to watch one more movie. This is during the inflation stage of that blastocyst. Now it has to escape from the zona pellucida because the outer cells of the embryo are what attach to the uterine wall. And it does it by squeezing through this little tiny hole. And I would argue that in this process, this is a point at which twinning could occur. If that embryo is simply pinched in half and each half receives some of the ICM, then you would have 2 identical embryos. And we've actually observed this happening in the laboratory, at least on 2 occasions. Here's one report where we see twin embryos. One of them is in the process of hatching, and these 2 embryos separated, and all 3 of them were transferred to the mom, resulting in a triplet pregnancy. So twinning can occur at this stage, and we don't need to imagine that totipotency exists in the 2-cell stage and that that's when twinning occurs. And certainly by the blastocyst stage, many, many, many events have established the identity of the blastocyst as a stage of the development of an organism. Okay, conclusions for twinning. The phenomenon of identical twinning was used to assert that early embryos were pre-persons or non-persons, and many philosophers also questioned whether the zygote could be considered a human being. The zygote is an individual human with the proximate active potential to undergo development, while individual cells are parts with only remote potency for development and must undergo a substantial change to become a human individual. So twinning is likely to occur at the blastocyst stage when the individuality of the original embryo has been firmly established. Okay, I think I am done with time. We're going to skip conjoined twins. It's only like 3 slides if you want me to go. Okay. Father Thomas has given me the imprimatur. Okay, so dicephaly or conjoined twins. Here is a conjoined snake with 2 heads. It occurs naturally in non-human animals, possibly due to overexpression of a particular transcription factor in early development. Anyone who's interested, it's probably TBX6. And we see it across the animal kingdom. So here in a reptile and an amphibian, and even in mammals, twin-headed goats. Human dicephalus is rare, but it does happen. So this is a 18th century skeleton displaying axial and head duplication. But this isn't simply a historical thing that happens. There are living cases of people who have dicephaly. So Abigail and Brittany Hensel were born in March of 1990 in New Germany, Minnesota. I was living in Minnesota at the time. And they're clearly distinct individuals with unique personalities, thoughts, and tastes. They share a single unified body that they jointly control. Philosophically, I would argue that they do not share a single soul. There's only one case known of a single nature being shared by three persons. But rather, they have two substantial forms that jointly and perhaps symbiotically uh, cooperate to support the life of both individuals. And disentangling which soul orders which aspect of the body may not be possible in situations like this. But it does raise some interesting philosophical questions. Even more challenging would be cases like this, craniophagus parasititis, which is, again, a rare condition. This is a case of a little boy born in 1783. His parents displayed him for money, and he was perfectly healthy, grew to the age of 4, died of a cobra bite, depriving the family of a source of income. But again, this does happen medically. In modern times, this was a little girl who had a head duplication. And the status of this duplicated head, typically they do not show complex behaviors. No one has ever, to my knowledge, actually recorded out of the brains of these duplicated heads to see what kinds of neural activity they have. They attempted surgery to separate her, but she died, sadly. So, conclusions regarding human nature. These are all things you've seen before. We have a zygote that is a living human being, exhibiting all the characteristics of a mature and albeit immature living organism. All properties of living beings are ordered by the soul. Soul is proposed to be an ordered relationship among parts. And twinning, whether it occurs at the two-cell stage or later, disrupts an ongoing developmental sequence. And conjoined twins represent a philosophically complex case of two souls informing a single organism. So with that, those thought-provoking observations, I'm going to stop.